Minimal residual disease, MRD, is becoming a very important tool in the management of myeloma patients. So we have currently two techniques, flow-cytometry or next-generation sequencing, NGS. And so in the IFM Cassiopeia trial, we did compare the two techniques. And so we found a very high concordance between both. However, I have to say that we always have a higher level of MRD when using NGS as compared to flow cytometry. And the main reason for that, I think, is that during transportation, which is usually 24 hours, some plasma cells are dying. And since flow cytometry is just analyzing variable plasma cells, and NGS is just looking at DNA. So DNA from plasma cells which are already dying, we are measuring it. So it is the reason why we have a higher level of MRD by NGS than with flow. I don't say that one technique is better than the other, but the conclusion of that is that at the patient level, you should always analyze your patient with the same technique. And if you use MRD as an endpoint, for example, in trials, you have to use the same technology for all the patients to be sure that your results will be comparable from one patient to another one. And so now, and in the next 1-2 years, I think we will use MRD to adapt the treatment. So far it is impossible to say this patient is negative, we stop the treatment. We have to wait for ongoing trials looking at that. But I'm pretty sure that if you are MRD negative for 2 years, now you can stop it.